In this lecture, we shall discuss Vernonia amygdalina's effect on cancer. Cancer is a disease in which some of the body's cells grow uncontrollably and spread to other parts of the body. Cancerous tumors spread into or invade nearby tissues and can travel to distant places in the body to form new tumors, a process called metastasis. Cancerous tumors may also be called malignant tumors. Many cancers form solid tumors, but cancers of the blood, such as leukemias, generally do not. The first article we shall look at in this lecture is titled "The Anti-Cancer Activities of Vernonia amygdalina extract in Human Breast Cancer Cell Lines are mediated through caspase-dependent and p53 independent pathways." The study aimed to understudy Vernonia amygdalina's anti-cancer effects in human breast cancer cell lines. Results from MTT assay revealed that Vernonia amygdalina inhibits or stops the proliferation of MCF7 and MDA-NB231 cancer cells in a time and dose-dependent manner. Furthermore, this study revealed that Vernonia amygdalina induces apoptosis in the two cell lines. Apoptosis is when cells begin to die off, when cancer cells die off. Vernonia amygdalina brings about the death of cancer cells in breast cancer studies conducted in this research. Given that approximately 70% of diagnosed breast cancers express ERA, a crucial finding was that Vernonia amygdalina inhibits the expression of ERA and its downstream player, AKT, highlighting the potential clinical significance of Vernonia amygdalina. Moreover, Vernonia amygdalina exhibits synergism when combined with doxorubicin, which is a medication that is used in treatment of breast cancer, suggesting that it can complement current chemotherapy. Overall, this study demonstrates the potential applications of Vernonia amygdalina as an anti-cancer drug for breast cancer treatment. The next article we shall look at in this lecture is titled "Recent Perspectives on the Anti-Cancer Properties of Aqueous Extracts of Nigerian Vernonia amygdalina." The article is published in the journal called Botanics, Targets and Therapy in 2016. According to the research, mounting evidence suggests that treatment with low concentrations of aqueous leaf extracts of the edible Nigerian Vernonia amygdalina plant arrests the proliferative activities and induces apoptosis in estrogen receptor positive, estrogen receptor negative, and triple negative human breast cancerous cells, and in androgen independent human PC3. Also in anthemic mice, Nigerian Vernonia amygdalina potentiates increased efficacies and optimizes treatment outcomes when given as a co-treatment with conventional chemotherapy drugs. Evidence of its noticeable cytostatic activities, ranging from changes in DNA synthesis to growth inhibition, mechanisms of inducing apoptosis in different cancer cell lines, and in vivo anti-tumorigenic activities, and chemopreventive efficacy reinforce the idea that Vernonia amygdalina can indeed serve as a treatment for breast cancer cells. And the article goes on to give us ways to prepare the extracts using pesticide-free fresh Vernonia amygdalina leaves and rinsed with cold water, soaked overnight, and then crushed to a mixture. Following filtering through clean white gaze to remove particulate matter, the mixture was filter sterilized, and the resultant crude aqueous leaf extracts of the edible Nigerian Vernonia amygdalina plant demonstrates anti-cancer properties when used in low concentrations to treat cancerous cells. The next article we shall look at in this lecture is titled Vernonia amygdalina Anti-Cancer Activity, Authentication and Adulteration Detection, published in the International Journal of Environmental and Resources Public Health, of Environmental Research, published in the International Journal of Environmental Research and Public Health. According to this study, evidence suggests that most chemotherapeutic agents are less effective as treatment in patients with estrogen receptor negative breast carcinomas compared to those with estrogen receptor positive breast carcinomas. Moreover, African American women are disproportionately diagnosed with ER breast cancer compared to their white counterparts. Novel therapies effective against ER breast carcinomas are urgently needed to ameliorate the health disparity. Previous reports show that low concentrations of water-soluble leaf extracts of a Nigerian edible plant, Vernonia amygdalina, potently retards the proliferative activities of ER human breast cancerous cells in vitro in a concentration-dependent fashion. So this research shows that Vernonia amygdalina leaf extracts retards or stops the growth of ER plus human breast cancerous cells in vitro. The next article we shall look at in this research is titled A Comparison of Tamoxifen and Vernonia amygdalina on the Human Breast Cancer Cell BT549. Tamoxifen is a medication used in the treatment and management of breast cancer cells. According to this article, Vernonia amygdalina, also known as bitterleaf, has been used to treat breast cancer in its native country, Nigeria, and has been commended for its result. This study has examined the anti-carcinogenic effects of aqueous and crude extracts of Vernonia amygdalina on human breast cancer cell line BT549. This study also compares the effects of aqueous and crude Vernonia amygdalina to tamoxifen on BT549. The data indicated that all concentrations of the crude extracts of Vernonia amygdalina decreases the growth rate of BT549 breast cancer cells. All concentrations of tamoxifen inhibited the growth of BT549 cells. Only 3 of the 6 aqueous extracts of Vernonia amygdalina demonstrated some inhibition of BT549. Further tests are needed to determine if Vernonia amygdalina induces apoptosis, which is cell death. Clinical trials are also needed to demonstrate that the Vernonia amygdalina is similar or more potent than tamoxifen. So we see tamoxifen applied to a group and Vernonia amygdalina leaf extracts applied to the study as well. In comparison, the data indicated that Vernonia amygdalina decreases the growth rate. All concentrations of Vernonia amygdalina decreases the growth rate. Tamoxifen, however, stopped it completely. Vernonia amygdalina decreased it. So there is need for further research to know if more work is needed to bring Vernonia amygdalina to a place where it will stop the growth of breast cancer cells. The next article in this study is titled Proceedings of the Second Public Health. Well, is published in the Proceedings. The next article in this lecture is titled Anti-Cancer Activity of Vernonia amygdalina Extract on YDR colon cancer cell line, published in the Proceedings of the Second Public Health International Conference. According to the research, colon cancer is the third most common cancer in men and second in women. Cancer treatments include chemotherapy, but some chemotherapy drugs are resistant. Vernonia amygdalina is one of the most widely reported plants that can inhibit the growth of various cancer cell lines. This study aims to examine the anti-cancer activity of Vernonia amygdalina extract on YDR colon cancer cell line. Ethyl acetate extract of Vernonia amygdalina indicate that it has very strong cytotoxic activity on YDR cells. Further studies are needed to analyze the mechanisms of ethyl acetate extract in inducing apoptosis, inhibition of cell cycle, and to observe the protein expression associated with it. So we see very strong promises of Vernonia amygdalina on this baseline or initial study to inhibit the growth of colon cancer. But the researchers note the need for further studies in order to gather more evidence, and even in order to gather more evidence to support the use of Vernonia amygdalina in the treatment of colon cancer. The last article we shall look at in this lecture is titled Vernonia amygdalina exhibits a potential for the treatment of acute promyelocytic leukemia. The article is published in the Global Journal of Advanced Engineering Technologies and Sciences, published in 2018. The purpose of the study was to explore the efficacy of a medicinal plant, Vernonia amygdalina, as a new targeted therapy for the management of acute promyelocytic leukemia. The results obtained suggest that induction of cell death, DNA damage, and cell apoptosis are involved in the therapeutic efficacy of Vernonia amygdalina. Because Vernonia amygdalina exerts anti-cancer activity in vitro, it would be interesting to perform clinical trials to confirm its effectiveness as an anti-cancer agent towards the treatment of this form of leukemia in patients.